Hello everybody, and welcome to today's webinar on streamlining access to RCSB Protein Data Bank APIs using Python. I'm Dennis Peel. I'm a scientific software developer at the RCSB Protein Data Bank based at Rutgers University. And I'm joined today by my colleague Ivana Truong, who will be co-hosting this event with me. So just to give you an overview of what we have planned for today, we'll begin with an introduction to rcsb.org, the APIs that underlie it, as well as the RCSB API Python package that will become the focus of today's webinar. I'll then hand it off to Ivana to provide a hands-on demonstration of using the Python toolkit through the use of a Google Colab Notebook. We'll then conclude with a summary, some resources and links for you, as well as some announcements. And then we'll open up the floor for Q&A. Just some general housekeeping before we get started. If you have questions during today's webinar, please use the Q&A feature provided on Zoom. We'll try our best to answer questions as quickly as possible throughout the webinar, but for those that remain unanswered, we'll try to address those at the end. Just to emphasize, today's webinar is primarily focused on the recently published RCSB API Python package for accessing rcsb.org APIs. So while I will try to give a brief overview of our APIs, if you are new to them, we encourage you to please check out our previous webinar for a more in-depth overview. And that webinar can be found at this URL here. And actually, if you go to pdb101.rcsb.org, go to our train tab and go to training courses, you can find all of our previous webinars here. And so the API webinar I just mentioned is highlighted here. And likewise, today's webinar will also be recorded and posted here as well. I can't give you an exact date on when that will be posted, but probably within 2 months. Last, we encourage you to please sign up to receive emails about any other future upcoming training events. All right, so with that, let's get started. So first, to provide a brief background, the Protein Data Bank was established in 1971 as the first open access digital data resource in biology, containing just 7 crystallographic structures of proteins. It has since then grown to house more than 232,000 experimentally determined 3D structures of biological macromolecules, owing thanks to the tens of thousands of structural biologists worldwide who submit their structures for deposition to the PDB archive. The PDB archive is jointly managed by the Worldwide Protein Data Bank, or WWPDB, which consists of the RCSB PDB in the US, where we're based, PDB-E in the UK, PDB-J in Japan, PDB-C in China, BMRB in the US and Japan, and EMDB in Europe. We are committed to ensuring that PDB data is fair, that is, that it remains findable, accessible, interoperable, and reusable. And we ensure that PDB data is made freely available to the public with no limitations on usage. The RCSB PDB web portal at rcsb.org provides a one-stop shop for 3D biostructure data, providing access to the more than 232,000 experimental structures from the PDB archive, in addition to more than 1 million computed structure models generated by AI and machine learning methods from AlphaFold-DB and Model Archive. In addition to providing access to this structural data, we also provide a rich collection of tools for searching, visualizing, analyzing, and downloading these data. Additionally, on a weekly basis, we integrate this data with annotations from approximately 50 external data resources, such as UniProt, SCOP-E, CATH, and more. And importantly, all these tools and data on rcsb.org are made accessible via a powerful set of application programming interfaces, or APIs. The 2 main APIs underlying rcsb.org are the Search API and the Data API. The Search API is what powers all search capabilities. So it allows you to perform a query throughout the archive and then fetch back a list of identifiers that match that query. The Data API, on the other hand, powers all data retrieval needs, where you provide a list of identifiers and you can get data about those structures. So to provide a quick schematic of how these 2 APIs work and how they can work together, shown here is how illustrating how the Search API could be used to get all NMR structures, which then returns a list of all the identifiers. And then you can provide that list of identifiers to the Data API to get a specific piece of information about them, such as the molecular weight. In addition to those 2 APIs, we also offer several other APIs that power rcsb.org. For example, we offer a sequence coordinate API, which compiles alignments between structural and sequence databases, a model server API, which delivers atomic coordinate data in a binary CIF format, a volume server API, which provides access to volumetric data from X-ray and EM structures, as well as an alignment API that computes 3D structure alignments between structures. So for purposes of today's webinar, though, we're going to be focusing just on these two APIs, the search API and the data API, not just because they're the two main ones that underlie rcsb.org, but because these are the two that are currently supported by the Python package that we'll be presenting. So these APIs are heavily dependent on the way that data is organized in rcsb.org, and this organization is important because it influences how you can search and retrieve data. So at RCSB PDB, data is organized based off the hierarchy of macromolecular structure, which is shown here. So at the very top level, we have an entry, which contains everything within a single structure. And within an entry, you can have one or more entities or unique polymer sequences or unique non-polymers. For every entity, you can have one or more entity instances or chains of that entity. So for example, you could have two chains of the same sequence, which would be two instances of the same entity. Biologically relevant groups of instances are called assemblies, and then the chemical components in a structure are the small molecules. So this hierarchy allows us to organize data based off of what's relevant for each hierarchical level. For example, information like the title and authors and citation of a structure belong at the entry level because that describes the entire structure. Whereas information like the different sequences that are a part of the structure and the type of those sequences, whether they're protein, DNA, or RNA, that belongs at the entity level. Furthermore, different chains can exhibit different binding sites or secondary structure, and so that belongs at the instance level, and so on and so forth for the other hierarchy levels. In order to uniquely retrieve data for each of these levels, we provide unique object identifiers for each of them, which are shown at the bottom here. At the entry level, these are indicated with a 4-character ID, in this case 4HHB. And note, recently we are extending these to 12 characters. So that's this extended format shown below. In addition, entities are indicated with a suffix of an underscore and a number. Instances are identified with a period and a letter, assemblies with a hyphen and a number, and chemical components are identified with a 3- and 5-character code. So I just showed how different types of attributes or data items appear at different levels of granularity, depending on what they represent. These attribute names and what level they belong to is based off of a set of schemas, which are accessible via these URLs here. Importantly, these schemas are what our APIs rely on for searching and querying data, and therefore they are also what our Python package relies on for searching and querying data. So to provide a quick set of examples, this table shows the translation of how an attribute or data item would appear in our advanced search menu, and then how the API in the backend is actually interpreting that. So if you were to go to our advanced search page and try to search for a structure based off a structure title, it would display as this on the advanced search page, but really what's happening in the backend is it's interpreting it as the struct.title. And so these schemas will allow you to explore that translation from the advanced search to the API end. So speaking of advanced search, before we dig into the details of the API, let me just highlight some of the features that our advanced search offers. First of all, at the top, you can choose to include computed structure models in your search, which gives you access to complete proteomes of interest. And this toggle switch is also available down here if you're using our advanced search menu. We also offer a variety of different types of searches, which I'll go into in a second. But I also want to highlight that you can adjust the type of data that you're returning. So you can choose to return either the entries or the polymer entities or assemblies. And if you choose to return polymer entities, you can actually group your results based off of things like sequence identity or uniprot accession code. So grouping results allows you to consolidate your result list so you don't have to sift through a larger list of individual entities. To cover the different types of searches that we offer are shown here. At the very top, we offer full text support search, which is like a Google-like search where you can search by keywords or phrases. We also offer attribute-based search, which I kind of just touched on related to the schemas. in which you can search for structures based on some property of those structures, such as who the author is, what method was used to determine the structure, and so forth. We also offer sequence similarity and sequence motif searching, which lets you search for structures based off of a sequence identity or amino acid pattern or regular expression. You can also perform structure similarity and structure motif search, which allows you to perform structure similarity searches at both a global and local level, in which the local level would let you look for binding site motifs. And last, we offer chemical similarity searching, which allows you to search for structures containing chemical components of a similar SMILES or INCHI string. So now let's take a look at how search is working behind the scenes. Basically, when you construct a search on advanced search, you can pick and choose whatever search service types that you want and combine them using these Boolean and or operators. So for example, we could take a full text search and combine that with a sequence similarity search and group them together using an AND operator. We call each of these subqueries a terminal node, and we call the group of these terminal nodes group nodes. Furthermore, you can also have groups of group nodes. So as you can see here, we have these two groups that are actually grouping together another group. So now let's take a look at how this is interpreted by the Search API backend. So then let's do so by just looking at this small snippet of the illustration over here, in which we have three different terminal nodes. We have an attribute-based terminal node, a sequence motif search, and a structure similarity search terminal node. And these two terminal nodes are grouped together using an OR operator, and then this group is grouped with this terminal node using an AND operator. So the way that this is interpreted on the API end is shown in this JSON syntax here. And you can see it kind of translates pretty directly. We see that we have three different terminal nodes indicated here, here, and here. And these two terminal nodes for the sequence motif search and the structure similarity are grouped together using an OR operator. And then this group is grouped together with the first terminal node using an AND operator. I'll also point out here that you can see this attribute-based search is using the schema-based value for that attribute rather than the common name that you'd see on the advanced search menu. When you execute the query, you then get a list of identifiers that match that query. So now let's switch gears to the data API. So in contrast to the search API, the data API supports the use of a GraphQL syntax for forming queries. Now, as a reminder, the data API is used to fetch data for a given list of identifiers. So as you can see in this GraphQL query here, we're requesting data for these three entries, and the data that we're requesting is below. So we're requesting the identifier for those structures, as well as the entity feature data, which contains annotations at the sequence level for those structures. So importantly, as described earlier, data is stored in a hierarchical fashion or a graph-like structure, which makes it amenable to the use of GraphQL. So the way that you would structure this query is by following this graph-like structure shown on the right, where we begin our query at the entry hierarchy level, and then we can traverse down the graph down to the polymer entities level, and then we can request data specifically under that level. And in this case, we're requesting the feature data. Another important point I'll make now is that you can actually start your query at whatever hierarchy level you want. So you could start this at the entity level and provide a list of entity IDs, or you could start it at the instance level and provide a list of instance IDs. And you can do that. And if you do that, you can choose to avoid having to traverse down through the graph. When you execute the query, you then get your response in the form of a JSON structure. And obviously, this is just a small snippet of the results from this query, but you can see that we get information like the protein family annotation for the sequence, as well as hydropathy values at a per residue level. So now let's take a look at how the search and data APIs manifest themselves on rcsb.org. And we'll do so using this pre-constructed query I have formed here for structures of hemoglobin from humans. So I'm going to go ahead and click on that. And it immediately brings us to this search results page where we see different entries that match this search. And we see that we have a total of 354 structures returned. So before we look at these, let's scroll back up to the advanced search builder and see how this is constructed. So since we're doing an attribute-based search and we're searching for structures of hemoglobin from humans, we're going to use two different attributes to do that. In this case, we're using the uniprot molecule name at equals hemoglobin, and that the organism is Homo sapiens. So I'll also point out that we also have all these other attributes available for you to explore on your own. In addition, we're including computed structure models in our results. So we've toggled that switch here. So it's pretty intuitive on how this query is constructed on that advanced search page, but let's take a look at how this is interpreted on the search API end. And you can do so by clicking on this little gear icon here, which when I click on it, will bring me to the corresponding search API query that was used to perform that search in that advanced search page. So as we've kind of looked at earlier, we can interpret these two terminal nodes that we've created, one for each attribute-based search. And we'll note that the name that we're using here is from the schema as opposed to what was shown on the advanced search page. So this button that I just showed you, getting to this page, provides an easy way to figure out that translation logic from what it appears in the advanced search menu versus how you would use it in our APIs. So we see we have two different terminal nodes, and we're also including computed structure models here. And when we run this query, we get our list of identifiers. We also get a total count of 354, so that's the same that we just saw. And we also get an associated score with each result. And so this is like a relevancy score, and the higher the score means the more relevant that result is based off of these two terminal queries. And obviously, by default, this is ordered in descending order. So now if we wanted to manipulate this query, we could do so in this editor here. And we could just add another terminal node at the same level. And let's say if we wanted to add sequence, we could do so. But you can see that just trying to manually type this is quite complicated, and it also kind of requires a prior knowledge of what the different possible values are, as well as what the different parameters are for each search service type. So this is one thing that the Python package will be introducing greatly simplifies. It alleviates the need for the user to know necessarily all the default values to enter, as well as having to worry about how to structure this complex JSON query. Particularly if you want to like make this more complicated and add a whole bunch of more levels of nesting, this can quickly become a challenging task because you have to worry about where your brackets are, where your commas are, and so forth. All right, so now let's go back and take a look at the data API. If you scroll back down, we can click on any of these structures, and it'll bring us to what we call the structure summary page, which provides a summary of the structural information. So we get information like the title, the organism, the authors, as well as the method that was used. And if we scroll further down, we see a breakdown of the contents of the structure. So we see the different entities that are part of the structure. So we have 1, 2, 3 entities here. And this second entity has 2 different chains or instances. Furthermore, if we scroll further down, we have the list of chemical components that are a part of the structure. And if we scroll back up, we see these other tabs here. So we could click on annotations, and we see structural annotations about the structure. And furthermore, if we click on sequence, we can get residue level annotations about the structure. So we can get information like the secondary structure, binding sites at different positions, levels of disorder and hydropathy. And so I hope you can appreciate the extent of data that's available through rcsb.org. And what's very cool is that all this data I just showed you is actually available through our data API. In fact, that's what's used to display all the data shown here. So if you click on this data API button over here, it'll actually take us to the data API query that's used to produce all the data that's displayed on this page. So if you're ever not sure on how to create your own data API query, you can take advantage of this to produce a nice boilerplate for you to work from. So we'll click on this, and we'll see that we had this super long query here for all this type of data. And that's because there's a lot of data to display on the structure summary page. So when I click play, we'll get that data in response. And obviously this is too much for us to go through right now. And one thing that you can do is you can actually you can truncate your query to just get the data that you're interested in. So let's say we're only interested in the identifier for some reason. So we're going to go ahead and we'll delete this. And we can rerun our query and we get specifically that data. Additionally, we could add another field if we were interested in. So for example, we used the entity feature data earlier. So let's go ahead and add that. And so in order to do that, we need to traverse through the graph. So we need to first go to the entity level. So I'm going to type in entities and we'll create a new nest here. And then if we search for feature, it should show up. And then let's grab the name from that feature. And so I can click play, and now we get the different entity features that are available for the structure at the entity level. And if you remember, there were 3 different entities on that structure summary page, and so we see 3 different sets of entity features that are returned, 1 for each entity. And if you're not sure where this attribute exists and how to get there, you can always take advantage of this documentation explorer over here. So every data API query begins at the query node. And so we can click on that, and that'll bring us to the list of hierarchy levels that we could start our query at. So we here started at the entry level. So let's scroll down to that. And if I click on that, it brings me to the core entry type, which I can click on. And then we can see what data values exist under here. And so we should be able to find polymer entities. So we can scroll down to polymer entity. Click on this, go to the core polymer entity type, and we can see the different types of data that are available under core polymer entity. And so one of these will be the feature, because that's what we were able to determine. And we can see that there's these different available data items underneath feature. So if you're ever not sure on where the data item is or how to traverse through the graph, we highly recommend taking advantage of this documentation explorer to do that. And you can also type in feature here and search for things that way. So while I'm sure you can appreciate the total potential of our data API for being able to perform very tailored queries for specific IDs and getting the specific set of data that you want, I'm sure you could also appreciate the amount of manual effort and kind of knowledge that you need beforehand to construct these queries. And so this is another thing that the API package that we're going to introduce helps alleviate. It helps automate the construction of these queries by not requiring the user to know necessarily the entire traversal down to the final data item that you want. And it'll also pre-populate data items underneath the final destination. So I know that sounds a little bit abstract now, but I think this will become clear with some examples I'll share in a moment. So let's go back to the presentation. And so with that, we're very excited to introduce our new Python package for facilitating access to rcsb.org APIs. This package is called RCSB-API, and the documentation can be found here. So the RCSB API package offers many capabilities to easily interact with our search and data APIs. And so to do that, it's currently organized into 2 different modules. We have a search API module and a data API module. The search API module lets you perform any type of search and get back a list of identifiers that match that query. The data API module lets you provide a list of identifiers and specific pieces of information that you want about them and get the data back. So while the package currently supports these 2 APIs, we do plan to extend support to the other APIs available at rcsb.org, and that will come with time. This work was previously published in the journal Molecular Biology, and if you're more interested in the architecture of the package, as well as more details about our APIs, I encourage you to check out this reference here. So to highlight some of the features and benefits of this package, I have listed here what each module can offer in terms of facilitating API access. The search API module, which enables fetching of structure IDs based off some search criteria, allows you to easily construct queries using an intuitive Boolean operator syntax, as opposed to this JSON syntax that's seen in the query editor. The search API module also supports all types of searches that are available on our advanced search page, such as full text search, attribute-based search, sequence and structure similarity, and so forth. It also It also offers another mechanism for providing your own custom structure files for performing structure similarity searches. So if you're generating your own structures using AlphaFold and you want to perform an archive-wide search for similar structures, you can do so by simply providing the path to your file on your computer to the package, and it'll perform that search for you. You can also include computed structure models in your search, and you can also group your results, such as by sequence identity, as well as performing faceted queries, which allows you to aggregate results based off of some attribute. The data API module, which enables the retrieval of data for a given list of structure IDs, provides several improvements for facilitating a query construction. And one of those is that it helps automate the resolution of partial and nested field paths. Again, that might sound abstract at this moment, but in the example I'll share in a second, you'll understand. And so another benefit that this offers is it provides an easy mechanism for fetching data for all structures. So let's say you're interested in the citation information for all structures in the archive. This offers an easy tool for getting that information, as opposed to having to first provide the complete list of all structure IDs. Last, both modules provide a set of helper functions for exploring our schemas and discovering attributes. So to illustrate how the package facilitates usage of the search API, shown here is the same query that we showed earlier. And you can see that this was constructed using 3 different terminal nodes and 2 group nodes. And this is the API JSON syntax. Well, this entire query can be reduced down to this small Python snippet shown here at the bottom, in which you simply need to create each terminal node as its own variable. So here we're creating an attribute query terminal node, a sequence motif query, sequence motif terminal node, and a structure similarity search terminal node. You then can group these terminal nodes together using a very intuitive operator syntax. So you can use the and or operators from Python in order to group these terminal nodes together, alleviating you from having to create these nested JSONs. Then you can get your results by just executing the query and putting them into a list. Similarly, in the case of the data API module, this query that we showed earlier can be reduced down to this small Python snippet showed up here. So instead of having to specify every single data item underneath the entity feature level, you simply need to provide RCSB Polymer entity feature, and the package, which knows about the GraphQL schema that's working behind the scenes, can automatically construct this query for you. And it'll pre-populate all these data items underneath your destined data target, and it'll also figure out the traversal path down to that level. So this greatly simplifies the ability to construct data API queries. Finally, these 2 modules are best used hand in hand, in which you first use the search API to find structures according to some query, and then use the data API to get data about those structures. So shown here is a real use case in which, if you're interested in getting all the binding sites across all structures for the HIV protease inhibitor ritonavir, you can follow this pipeline by first starting with a search for all structures that contain ritonavir, and you get up in response a list of all identifiers. You then provide that list of identifiers to the data API module and request the instance feature information from those structures. In response, you get the list of instance features, which contains information like the ligand interactions, as well as the specific binding sites of those ligands. And so you would then parse this result set and extract out the specific information that you want and adjust it to however you need for your own research purposes. And actually, Ivana will be demonstrating this pipeline upcoming right now. So with that, I'm going to hand it off to Ivana to provide the guided demonstration of our RCSB API Python package.
Thank you very much. So let's get into the live demonstration. So here is the GitHub repository where all these Python notebooks will be posted. And then once we get to that repository, we can open each of these notebooks in Google Colab, which should open the notebook right in your browser, and you can access it as long as you have a Google account set up. So here you can see, here are all the Python notebooks that we'll be walking through today. But actually, I'm going to start by bringing you to our documentation and showing you that. So here's the installation information. And then you can see that our documentation is split into two halves, the search API half and the data API half. They're both organized very similarly, so I'm just going to go through the search API portion first. Or we're just going to go through the search API portion right now, and you can explore later. The quick start will just get you the very basic information for starting with simple queries. Then you can go to query construction for more complex examples and different options you can use. Exploring schema attributes would be similar, where there are different methods that you can use for finding different attribute names. Additional examples shows all the different query search types that you can use and how you would use them. An API documentation shows the doc strings that are associated with each class and all their methods as part of this package. Next, I'm just going to bring you to the GitHub. This is our GitHub repository for the Python package. And so all the code that makes up the package is posted here. If you want to go and see how it works, you can definitely explore this. If you have any issues, you're definitely welcome to report them, or if you wanted to make any contributions as well. So let's get into the demonstration. We're going to start with exploring the Python search API portion, and then we're going to explore the data API portion of the Python package. And finally, we'll see how we can use the search and data API together in a workflow. So once the notebook loads like this, you'll see there's this open in Colab button in the top left. So you can just click that and the notebook will open in Google Colab. We're just going to make sure that the Python package is installed in this environment. So in the search API Python package, there are many different search types, but we're going to start with the two of the most common ones. So we're going to start with text query and attribute query to search for PDB IDs associated with the phrase hemoglobin and from Homo sapiens. So we're going to start with importing text query and attribute query from RCSB API.search. We'll use text query to search for structures associated with hemoglobin. That's that Google-like search that Dennis was mentioning. And then we'll use attribute query to search for structures with Homo sapiens as the source organism. So we'll find this RCSB entity source organism attribute, and these attribute names are defined by the schema, like Dennis mentioned. We'll use the exact match operator, and then there's other operators as well, like contains phrase or exists, and then other attributes will have different sets of operators that are valid for those. We're going to set the value equal to Homo sapiens. Once we've created these individual queries, we can combine them using these operators. Ampersand indicates and, type indicates or, and tilde represents not. So we're going to combine these two queries using the and. So we're searching for structures associated with hemoglobin and with Homo sapiens as a source organism. So we've created that query here. To execute that query, we're going to run it as a function. So we've added these parentheses. And the results are returned as an iterator of result identifiers. So we can iterate through them in a for loop like this. You can also just convert the results into the list and then work with it exactly the same way as you would any other Python list. So here we're slicing it to see just the first 10 results. It's important to note that by default, queries are only going to return the IDs of experimentally determined models. You can control whether computed structure model IDs are returned through the return content type parameter. So we're going to use the same exact query as above, except this time when we're running it, we're going to add this return content type parameter, and we're going to include computational and experimental IDs. So you could set that to be just computational or just experimental, which would be the default option. We're just going to look at the first 10 results here. And you can see here that there's some CSM IDs in the results now. So as we've mentioned before, there are many other types of searches besides text query and attribute query, and these are all supported by the Python package. So here on the left, You can see these are the search services that the search API offers. And then on the right, these are the equivalent query types in our Python package. These different search types can be mixed and matched for more specific results. So below, we're going to use a sequence similarity search and an attribute search to identify polymer entities that share 90% sequence identity with GTPase HRAS protein from humans. Then we'll use the attribute search to exclude structures that have more than one mutation. So we'll start by importing attribute query and then sequence similarity query. We'll build the sequence similarity query here by passing in the sequence we would like to search for. And then we're going to set identity cutoff to 0.9 because we want our results to share 90% sequence identity. And then next, we're going to use the attribute query to limit our results to structures with few or no mutations. So we're going to use this RCSB mutation count attribute and set that less or equal to 1. We're going to combine these individual queries with an ampersand to indicate and. And then we're going to run that query with return type polymer entity, since sequence similarity search needs to be run on polymer entities. And we're just going to look at the first 10 results here. So you can see these are the different polymer entities that match with our search criteria. Another useful search type for comparing structures is the structure similarity search. And you can use structure similarity search with entry IDs, file URLs, or local files, but I'll just show you how to use file URLs here. We'll start by importing struct similarity query, setting our search type to file URL, passing in our file URL, and then specifying that our file format is CIF. And we'll show the first 10 results here. And you can also search using a local file, and you can see our read the docs for examples of how you would set that up. And for examples using these and all the other search type, you can also check out our read the docs. So next, I'll talk a bit about faceted queries. Our search API and the package also support facets, which will sort the results into buckets based on the returned values. And this allows you to calculate statistics for your results and kind of get that higher level view. So for example, you could search for how many models released since 2000 were determined by each experimental method. So we'll start by importing attribute query and facet and creating an attribute query to find all the structures released after 2000. And then when you're executing that query, you can add a facet to this facets parameter. You can set the name to anything you want. In this case, experimental methods is what I set it to. Aggregation type is terms, and for more specifically what terms means, and then the different aggregation types that are available, you can check our documentation. And we'll be sorting or aggregating our results based on what is returned for experimental method attribute. To access the facet results, you can use this results object and then access the facet attribute. So here you can see on the left side, these are the different experimental methods and the number of results, the number of entries that are determined using each of those experimental methods. So for more information on facets, you can check out our search API documentation or our Python package documentation on Read the Docs. And you can find additional examples using other API features like grouping results or sorting results based on different scoring on our Read the Docs as well. Once you've constructed a search API query, you can take a look at the full query in our search API query editor. So here we're going to construct a brand new query. We'll start by importing text query here and then importing search attributes as adders. And this search attributes object is just another way you can make attribute queries. Whether you use the attribute query class or the search attributes object, they are equivalent and will give you the same results. So here, let's start with using text query on the string interleukin. Then we'll use the adders object to search for source organism of Homo sapiens and drug groups investigational and experimental. Once we have these individual queries, we're going to combine them using and or operators. So in this case, we're searching for a structure associated with interleukin and from the source organism Homo sapiens and has an investigational or experimental drug group. So let's run that here. So once you have that completed, you can use the get editor link method on the result. And you can see this is the raw query that the Python package has generated, and you can run the query in the Search API query editor as well. Another little feature, if you use the search attributes object, is that you can have tab completion or auto completion. So you can see here that if you start typing an attribute name, it'll give you the different options of attributes that you could search. So as you might have noticed throughout this example, there's a lot of different attributes and attribute names, and it can be difficult to know where to start or what attribute you intend to search for. So there's a few ways to explore that through the package. We'll start by importing search attributes again, and then using this search method, which you can pass in a string or a regex pattern to find matching attributes. So here you can see these are all the attributes with ligand in the name, and then their descriptions here. Or if you already know the name of a specific attribute and you just want to find just those attribute details, you can use get attribute details on one attribute here and find just that information. If you're looking for resources outside of the package, you can also look at our Search API documentation, where information is split into the structure attributes and chemical attributes. If you've built a query in the advanced query editor and want to know the corresponding Search API attribute name, you can also check this attribute details page. And of course, for further documentation, you can reference our Read the Docs page. So next, let's go to the Data API notebook. And here you can see the Open in Colab button again. So we're just going to open that up in Google Colab. We're going to make sure that the RCSB API package is installed. So in the Data API portion of the package, there's just one type of query as opposed to the Search API. And there's just a few arguments that you'll need to create that query. So first is input type. This defines the starting point of your query. Some examples include entries, polymer entities, or chemical components, chemcomps. If you're unsure of which input type to choose, you can usually use entries and access the other input types from there, but there are a few exceptions to that. The next argument is input IDs. These are the identifiers of the given input type that you would like to search for. And there are specific formats for PDB IDs of different input types. So here's the format you're probably most familiar with from the website for entries. And then there's different separators and numbers or letters for different input types. Finally is return data list, and this is the data you're going to request for each of the given input IDs. So let's create our first query. We're going to start by importing data query as query from rcsb.data. And we're going to use the input type entries, input ID of 4HHB. And then for return data list, we're going to use experimental method, exptl.method. So once you've created that query, you can execute it using the exact method here. And the response is returned by that exact method. So you can see this is the JSON response. You can also access the response through the object. So query.getResponse will give you the same exact response. So by using the Search API and Data API together, you can first refine a list of IDs that are of interest to you, and then request data on those particular structures. So we'll go through a quick example, and then we have a notebook after this as well. So in this example below, we're selecting human structures associated with the phrase hemoglobin, or associated with the phrase interleukin with investigational or experimental drugs. And this is the query that we created just now in the Search API notebook. So once we've narrowed down our structures of interest, we're going to request the structure's experimental method and resolution. So here, this is the same query we just created in the Search API notebook, so I won't explain it too much. And then we're just going to save the first 50 IDs within ID list here. And then we're going to create our data API query here. So for query, we're going to use the input type entries. And then for input IDs, we're going to use ID list, which is that first 50 IDs returned by the search API query. And for return data list, we're going to search for experimental method and then the resolution here. So we'll execute that and then look at the results. And you'll notice here this warning is returned. What that's saying is basically the Python package is dynamically creating parts of your query, and that if your code is meant for long-term use, it'll be more stable if you just use these fully specified paths here. So you could just copy paste this into the return data list argument here, and that'll give you the same results, but it's just more recommended for long-term use. So here's the response, and you can see that the RCSB ID was returned, the experimental method, and then the resolution as well. So if you're not interested in narrowing down your structures of interest, but are actually interested in the archive-wide data, you can use all structures to request fields for every entry or chemical component in the PDB. And just know that these queries are going to take longer than if you were to query using a more refined list. So we'll start by importing all structures from RCSB API.data and then importing data query. We're going to set input type to chemcomps and then use all structures as our input IDs. So since we set the input type to chemcomps, this is going to search for all chemical component IDs in the PDB. And then for return data list, we're going to search for the corresponding drug bank ID if that exists. When we run this query, we're going to add this progress bar equals true parameter, and this can be added to any query, but it can be helpful if you're running particularly large queries to see the progress of of the requests. And then we're just going to look at the first 10 results. And commented out here is an example of using all structures with entries. So in this case, it would be searching for all PDB entry IDs, and then we would look for experimental method. I commented this out just because it takes longer to run and is pretty resource intensive, but you can certainly come back to this notebook later and run that if you would like. So here you can see these are the results and the different drug bank IDs if they exist. So like the search API part of the Python package, once you have a query constructed, you can visualize it in our data API query editor. So we're going to create this simple query here, and then you can use query.getEditor link to see the editor, to see the query in the data API editor. So here's the query that was generated, and you can run the query and then explore the documentation as well. Similar to the search API portion of the package, there are also ways of exploring the schema within the package. So we can start by importing data schema and then initializing a schema object here. And then we're going to use find field names with a string to find all the different fields with that string in it. So here, these are the different fields that we could use. Once you find the field that you would like to search for, you can find a path to that field by putting in your input type and return the name of that field into find paths. So here from entries to RCSB ligand neighbors, you could copy paste any one of these paths, and it would take you from that input type to this field name. You can also explore a schema through the data API query editor's documentation explorer, which Dennis showed. And for further documentation, you can always reference our Read the Docs page. So finally, we're going to go through our search and data workflow notebook. And this shows you how you can use the search API and data API together. So as we've been saying, the search and data APIs are most powerful when you use them together. And you can use the search API to identify structures of interest and then use the data API to request information about those structures. So in this example below, we're going to use the search API to request structures with the HIV protease inhibitor ritonavir bound. And then we're going to identify which residues interact with ritonavir using the data API. And this is that example that Dennis briefly mentioned at the beginning. So we'll start with the search API query. We're going to start by importing search attributes as adders, and then use this different identifying information about ritonavir to find all the structures with ritonavir in it. we're going to use and then look at the first 10 results. So these are all structures with ritonavir. Once you have this list of structures, you can request data on each structure's interactions with ritonavir. So import data query as query, then we'll set input type to entries. We're going to use these search API results as our input IDs, and we're going to search for RCSB polymer instance feature, which contains all the ligand interaction information. And then we're going to search for the instance RCSB ID as well. We're going to execute that and look at the first result. So here you can see all the instance information and the RCSB ID as well. When you have that JSON response, so all data API queries are going to get that JSON format response. Once you get that response, you can parse it into a format that's most helpful for you. So in this code below, we're going to parse our results into a nested dictionary where the keys are instance IDs and the values are lists of ligand interactions and information about that. I'm not going to go too much into this code because it's really going to vary depending on your use case and what you're looking at, but you're welcome to revisit this notebook and look through it if you would like. So here you can see this is the instance ID and then different information about the interactions and the residues that are interacting. So this example is just an outline of a general workflow of using the search API to identify structures of interest and then using a data API, our data API, to find more data about each structure. And then finally parsing that data API response into a data structure that suits your use. And you can see how this has a lot of applications for a lot of different types of projects. And as always, for further documentation, you can always look at our Read the Docs page. And now I'm going to pass it back to Dennis to wrap it up and then open our Q&A session.
Great. Thank you very much, Ivana, for that wonderful tutorial. We're now just going to conclude with a brief summary, some resources, as well as some announcements. So to summarize, the RCSB API Python package enables facilitated access to rcsb.org search and data APIs, which helps increase the fairness of PDB data. All source code is open source and available on GitHub at this link here. And so we encourage you to, if you run into any issues using the package, please report them there, as well as we gladly welcome any contributions. And you're welcome to use this package however you wish in your own research purposes. Furthermore, Ivana just demonstrated the usage and applications of the package through the use of some Google Colab notebooks. Future directions that we have planned, we intend to add support for additional rcsb.org APIs, such as the sequence coordinate API, as well as adding support for the downloading of 3D coordinate file data. And this would be taking advantage of our model server API. I also wanted to provide a single snapshot of all the resources and links that we've referenced throughout today. So if there's one screenshot you want to take, now's the time to take it. We'll also copy and paste these into the chat for your own reference too. But basically this just provides links to our Python package documentation, the GitHub project, the webinar notebooks that Ivana just demonstrated, some information about our APIs, and where our webinar recordings will be published. Additionally, as I talked earlier about our schemas that are used for defining these different data attributes, I wanted to provide those references here too for your own exploration. And with that, I'd like to acknowledge the entire RCSB PDB team for all their contributions to every aspect of our data resource. Of particular note, we'd like to thank our director, Dr. Stephen Burley, and our associate director, Christine Zardecki, for their leadership of our organization. And I'd also like to thank Brenda Vallet for her support with project management of this project. In addition, we'd like to thank the Research Intensive Summer Experience Program, or RISE program at Rutgers, through which much of this work was performed, particularly by Habiba Morsi, Santiago Blauman, Ivana Churang, of course, and Rusham Bhatt. And last, we'd of course like to thank all of our funding agencies, without which none of this work would have been possible. So a special thanks to the U.S. National Science Foundation, the National Institutes of Health, and the U.S. Department of Energy.